So we were talking about B cell membranes. And remember, there are membranes on the top of these cells, like B cells. And these membranes have receptors. And these are the receptors that will take the signal. So these are the receptors with which the B cell will talk to other cells. Okay? Now, let's talk about what are those signals and how do we manipulate them. But before we manipulate them, we need to know what are they and what are their functions before we go and want to mess up with these signals. Now, if you pay attention to this slide, these are some of the important molecules expressed at the surface of B cell. So remember, as we are going on in the first phase of immunology, basic immunology, I'm going to talk about the major cell surface receptors. And as I go in the lecture, I'll also point to you what are the important, and you can make note of it, and then they are worth remembering. Now, in this case, there's a B cell surface, as you can see from here, and then these are different molecules, the receptors. And the most important receptor the B cell has on top of it, which will act as a fragment for antigen binding or antigen capturing thing, you will see that is a BCR. So this is a BCR, a complete BCR, B cell receptor, which is an immunoglobulin gene, and looks like an antibody, which has a heavy and light gene. And then again is attached by alpha beta Ig transmembrane proteins. Now, this is the receptor that basically B cells use for capturing of antigen. And then again, anything that associates with this molecule will act as a transducer, and it will signal the events that need to take place. So this message that comes from outside has to be taken from there. Then there is something called co-receptors. So you see B cell has co-receptors. These are additional signal transduction molecules, and they're also important. And right now, the most important for your course are, again, cluster of differentiation CD, and you can see 29, 19, and 81. So these are different type of proteins, and these are the receptors that are embedded in the cell membrane or B cell membrane, and some of them, of course, have an external and of course, they will communicate to inside as well. Now, of course, one of the important functions of B cell is that B cell talks to T cell. So B cell and T cell lock in. So what is the docking site? So they have some of the molecules mentioned over here. There's a list of that. The most important one is that they have major histocompatibility gene complex, especially class 2. And today, after these couple of slides, I'm going to talk about MHC, major histocompatibility class 1 and 2. But remember for now that 2 are important, 1 and 2. B cell expresses 2. And this is a receptor that binds the peptide and basically interacts with TCR of CD4. The CD4 is a T cell, and the B cell, they will lock in where the T cell receptor will lock in with MSC. Then again, like co-receptor, we have co-stimulatory molecules. As the name suggests, they will stimulate an action. So for example, if you want to send a signal to a B cell, we need to make antibodies. So we need to control that. We need to either expedite or stop or reduce or slow down. So this is like a braking action we have via co-stimulatory molecules. And the most important is CD40, B7, and ICOS or ICOS. So these are the three important docking sites that B cell has, and the other cells will come and dock in. And once these two molecules will interlock, then they have very specific action. For example, if you lock in with CD40, it has a corresponding, remember there is a receptor and there is a corresponding receptor. So the receptor expressed on B cell is CD40, and the ligand expressed on another cell will be CD40L. So that's a ligand for that, and this is an important switch to turn on isotype class switching. So the question is, which door you want to open to let class switching take place? So the key that you need is CD40. Okay? And then we have B7, which is very important. And this interacts with another molecule that is expressed on the other cell. We'll talk about that in detail. And finally, there is a ICOS ligand that acts on activated T cells. So you can see from here, the whole idea is that since we're talking about the players, and we're talking about some of the molecules expressed on B cell, so if you know what is expressed on B cell, you can pretty much determine what are the sites available. So if you like 5 fingers over here, 5, so you can see like interdigitating effect that will take place. Now, just to give you an idea that B cells exist when they are formed in bone marrow as naive cell. This means they are naive. They haven't seen any antigen. Once they leave bone marrow on demand or per need, then what will happen is they will become activated. So there is an action item. There is a cell which is resting, and there's a signal that's going to activate this cell. So we'll talk about B cell, activated B cell, a T cell, an activated T cell. Now you can see from here that this is a B cell, and B cell has a receptor, which is a B cell receptor with the Ig molecule. And what happens is that one of the bacteria comes over here, and this bacteria has an antigen, and this antigen is picked up by B cell receptor. Okay? Now signal transduction will become, and signal will pass through this B cell receptor to give a message to B cell. For example, we need to make antibodies. Okay? Now remember I just said that some of the co-receptors are there to enhance this effect. So you have a first dose, and you have a second dose. The second dose is to enhance the first one. Okay? Now in this case, what happens is that if you remember your complement system in physiology, so complement was a very complex series of plasma proteins. So one breaks down into 2, and then so on and so forth. And then you are left with these tiny little particles. They are C3 proteins, and when we go for blood tests, we look for C proteins. So this means that there is an activity taking place in the blood where your system is producing complement. So this complement, especially C3DG, just remember complement protein. So this will again bind to this bacteria. And interestingly, this complement basically is a receptor for a co-receptor expressed on top of the cell. It's kind of an overdrive. So what will happen is that it will enhance the signal. It will speed up the process. Because remember, we are looking for all the ways that are possible for us to make a good immune response. One of the good immune responses is that we are infected, we have a challenge of bacteria, right? And we need to fight bacteria. and B cells are there to help. So this is something that we learned from B cells, that how do we enhance the signal through BCL. So remember for now, the co-receptors are there, and when they get engaged, they would enhance the function of B cell. Okay?